These two snakes are siblings. They came from the same clutch and they are brother and sister. Same mother, same father. One is male and one is female. How does that happen?
The visual appearance of these snakes is quite different, and you'll often hear it said that one of them has more genes than the other, and that isn't true. They both have the same number of genes. They both have inherited a full set of chromosomes from their parents. They must have, because they're perfectly formed and easily identifiable as ball pythons. So whilst these animals have the same number of chromosomes and the same number of genes as their parents and as each other, they have actually received different alleles from the parents. This means that they have inherited mutations from one or both parents, and one of them has received more genetic mutations than the other one. Neither of them has a full set of genetic mutations from both parents.
How does that happen? This baby here came from two quite ordinary looking parents, one of which was yellow belly and one of which was asphalt, neither of which look anywhere near as spectacular as the offspring. How does that happen? This guy here is a multi-gene banana. His father was a male banana, he is a male banana, and all his banana offspring are more than likely also going to be males. How does that happen? This little girl here was born kinked. You can see she has a deformity right behind her head. She also has a little bit of stuck shed, which we'll sort out in a moment. And she has another kink just here, about 2 inches down her neck. Why was this baby deformed when all its siblings were perfect? How does that happen? Hi guys, it's Rob from Royal Balls. Here we are with the genetic video that I've been promising, and we're going to cover rather a lot of ground. We're going to cover terminology, we're going to cover meiosis versus mitosis, we're going to cover temperature sex determination, we're going to cover parthenogenesis, and we're going to cover genetic deformities. So the video has turned out to be quite a piece of work. It also means that I'm having to split it into 3 parts, because it also turned out to be much longer than I anticipated. So part 1 is going to be terminology and Joe Tully's section, which is well worth watching. Part 2, we're going to cover mitosis versus meiosis, and specifically, is temperature sex determination in our ball pythons a thing? How does that work? Part 3, we're going to cover parthenogenesis. Parthenogenic clutches, I think, are becoming more widely recognised. How does that work at the cellular level, and how do we recognise a parthenogenic clutch? And we're also going to look at genetic deformities. A lot of genetic deformities are attributed to incubation temperatures. And yes, incubation temperatures can cause problems, but a lot of problems are actually genetically predetermined. How does that work? We're going to take a look at genetic deformities as well in part 3. So here is part 1, and it gives me great pleasure. I am honoured to have Joe Tully help me produce this video in collaboration. So in part 1, which is the part you're about to see, we are going to cover some basic genetic terminology, which we will need moving forwards, and Joe Tully's magnificent contribution, where he analyses a deformed snake, talks about how that deformity came about, and also manages to save one of the snakes. This footage is going to be extremely valuable, and it gives me great pleasure to incorporate that into this part 1. So without further ado, let's get after it. Okay guys, let's start off with some basic terminology. Everybody has seen my Punnett square and my genetic calculations for pairing outcomes, and I think everybody understands that concept, and it is actually much simpler than people think. So trust me on this one, this is going to be even simpler. We do not need the Punnett square. All we need are the pipe cleaners and our genetic mutations for this demonstration, and I'm using black here to represent male and pink to represent female, but in this first bit, which we're going to build upon later, it doesn't really matter at the cellular level. So everybody is familiar with me saying that we split the genetic material of the female in half and we split the genetic material of the male in half, and we produce sperm and we produce eggs. So we take half of the male's genetic material and pair it to half of the female's genetic material to produce the new offspring. So every single ball python that we have has got genetic code derived from mum and derived from dad. Now, this genetic code is actually a DNA strand. This DNA strand contains genes, and these genes are the genetic code that control the production of proteins and hormones during the development of the embryo. The genes actually control the timing, the switching on and the switching off of various protein substances and hormones, and it all has to happen in concert at exactly the right time to produce the offspring. So, as you can imagine, this DNA strand is actually quite complicated. It's a double helix twisted around itself, as you've seen in my DNA demonstrations, but nature has actually made it a little bit easier to handle because this genetic strand, this DNA strand, contains so much information. Your body's broken it down into pieces, and those pieces are called chromosomes. There are 18 of them in a ball python in a full set. So your ball python offspring receives 18 chromosomes from dad and 18 chromosomes from mum. So let's drop down a level. Instead of looking at the whole DNA strand, let's just look at a single chromosome. Remember, there are 18 of these, so everything that I say about one chromosome is going to be mirrored for the other 17 chromosomes. So in total, you have two sets of 18 chromosomes, or 36 chromosomes. Because each chromosome has a corresponding chromosome from the other parent, you are actually receiving two copies of the same gene. Along this chromosome here are hundreds, thousands, millions of genes, each one occupying a particular slot, and it is mirrored on the other side from the other parent by exactly the same gene. So each side has exactly the same number of genes, and the corresponding location for that gene on each of the chromosomes is called a locus, and this gene and this gene are called alleles. So you have exactly the same number of genes in each chromosome, but if a gene is modified and carries a genetic mutation, it is known as an allelomorph. So here are our genetic mutations which reside at the location of the gene at a particular locus. Each of these sides here is known as an allele, and if we get only one copy of the allelomorph, it is obviously heterozygous. Now note that the other side is not empty, it contains the same gene, just an unmodified version of it. So here we have the heterozygous form of that genetic mutation. There are always two slots because there's always a corresponding chromosome from either parent. So there are two chromosomes, two slots, so we always have the possibility of that genetic mutation existing on the other side of the locus. And when they're the same on both sides are carrying the genetic mutation, this is the homozygous form of that genetic mutation. So here we have non-modified genes. It isn't true to say that it isn't carrying the gene. It is carrying the gene, it just isn't carrying the modification of that gene. Here is the heterozygous form of that modification where it exists on one side. So we've got two different alleles now across that locus, and here is the homozygous form of that genetic mutation. So we talk of genetic mutations as being dominant, incomplete dominant, or recessive, and actually that's an artificial division because there are two available slots on each side. Every single type of gene can exist in its normal form, in its heterozygous modified form, or its homozygous modified form. We tend to think of dominant genes as being the complete visual expression of that genetic mutation when this pair of chromosomes carries just one copy of that genetic mutation. Sometimes we get a visual expression, but it isn't the full visual expression, and those are the incomplete dominant genes. And sometimes we call the genetic mutation recessive, which means that one copy has no visual expression, and that in itself is not accurate because every genetic modification, even with only one copy, is going to do something. It's just that we don't always see it. In the homozygous form, if it's a dominant gene, the homozygous form makes no additional change to the phenotype. There is no difference in the visual appearance. For an incomplete dominant, the homozygous form would be the super form, and for a recessive, the homozygous form is necessary in order to see the visual expression of that genetic mutation. Now, note that in our ball pythons, in our designer morphs, we are dealing with colour and pattern mutations. But all of these genes carry various mutations which may or may not have an expression in the animal. They could actually cause problems during the development of the embryo. They're not all colour and pattern mutations. Some of them are for all sorts of different functions and different organs within the body. So when we work with these genetic mutations, we are just looking at colour and pattern. Now remember that this is just one chromosome, and there are 18 pairs of these chromosomes on the DNA strand in ball pythons. Each of these chromosomes is therefore the same. It carries the same number of genes in the same configuration as the parent, and that means that these two chromosomes here are homologues. They form a pair. Because these are paired, they are known as diploid. There are two versions of the same chromosome and two versions of all the genes that that chromosome contains within this homologue. For humans, there are actually 23 chromosomes making a diploid total of 46. We're going to come back to that a little bit later. Remember that these animals are XY animals, and one of the chromosomes inherited from each parent will carry the sex chromosome, either X or Y. A male animal is an XY and a female animal is an XX. This is important. We're going to come back to this later, but what that means is that actually the 18 pairs of chromosomes within a ball python, only 17 pairs are actually homologues or autosomes. One pair of chromosomes is actually slightly different. It could be X and Y, and again we're going to come back to that later. Okay, are you with me so far? Okay, let's just quickly cover the example I showed in the introduction of a snake that is carrying a copy of a different gene from each parent. If you remember, I showed a freeway which came from a yellow belly to asphalt pairing, and typically we call this allelic. What do we actually mean? Well, Dad was heterozygous for asphalt, and we know that asphalt can also exist in a superform. So here is the heterozygous version of asphalt, here is the homozygous version of asphalt. So this would be a super asphalt. Dad was an asphalt. Mum was a yellow belly. Yellow belly is a different gene but is in the same complex, and all genes in the same complex will reside at exactly the same locus on exactly the same chromosome. So Mum was a yellow belly, she was heterozygous for yellow belly. Yellow belly does have a superform where we get 2 copies of that genetic mutation in the homologous chromosome, and this would be an ivory.
What happens when we combine asphalt and yellow belly is that we get the 2 different genes, asphalt and yellow belly,
occupying exactly the same locus. So these genetic mutations, because they are modifications of the same gene, give you a wild interaction which is very different to the superform of either of the individual genetic mutations. Typically we call these allelic, but all genes that reside at the same locus are allelic, and this allelic version is exactly the same as a superform. So during cell division, when we create haploid sperm or egg cells, when we replicate this cell, we must get either a copy of yellow belly or a copy of asphalt. There will be no genetic material that does not carry the modified gene, either asphalt or yellow belly. Okay, so now we've got the basic terminology out of the way. You will hear some of this terminology in use by Joe. Let's get over to Tully's outrageous royals and take a look at some excellent footage.
Hello YouTube, I am Joe Tully at Tully's Outrageous Royals, and I am honoured and privileged to be welcomed on to Rob Barracross's channel at Royal Balls for a collaboration. We have quite a comprehensive topic to discuss, many topics actually, so without further ado, we're just going to get into the footage. I hope you all enjoy. If you've been following my channel, you may remember this egg cutting. we cut into this egg and we did actually have identical twins. 4 reasons that we're going to discuss in this video, because I was able to identify that these in fact were identical twins, just because of the fact that they were the same morph, and I could see the umbilicus deeper down in the egg and see that they were attached to each other rather than having a separate nutrition and separate blood supply. These were sharing a blood supply and nutrition, which meant they were identical twins, which meant I had to intervene and did what I could to save the healthy twin. My greatest fear being that because these are attached and sharing a blood supply, if the deformed twin was to perish, then that would also spell disaster for the healthy twin. The last thing you want in this situation is for the deformed twin to die and for the healthy twin to be trying to push blood round a dead vessel, essentially. So, for the purposes of identifying the different kind of twins that we might see in an egg, there are different kinds, believe it or not, and this is something that I think is quite widely misunderstood throughout the hobby. If I was not able to identify that I was looking at identical twins in my last egg cutting, then there's a very real possibility that we would have lost both the hatchlings. On the left hand side then we have identical twins or monozygotic twins. The key thing to notice about these is that they are conjoined by the umbilicus and the main vein which connects them to the blood vein network. The umbilicus also connects them to the nutritional reserves of the egg, so they share a yolk. Because these are identical twins, that means they will share the same chromosome, so XY or XX, so they will always be the same gender. They will also share an identical phenotype, so in my case both were burgundies, and that will be the same across any identical twins. They will always be the same morph. They share exactly the same genetic material. Which takes us nicely over to non-identical twins or fraternal twins. I tend to think it's a little bit easy to think of these as 2 eggs within 1 egg. They have a separate isolated blood supply and also have a separate yolk. I mentioned that these are just like 2 eggs within 1 egg, so for that reason these can also be of a different phenotype or a different morph and also be different genders. They can also be the same gender, it's important to note. It just depends on what chromosome is passed on through the male sperm. Just to extend on that a little bit further, it is possible to get the same morph within non-identical twins. The probability of that remains the same as it would throughout the rest of the clutch. And the probabilities for that are much more reduced if you have a higher gene stack within the parents. But to add another caveat onto that, non-identical twins can actually be dual sired if you have paired multiple males to that female, so the probability of that remains the same as well. Not to ignore the possibility of retained sperm from a previous pairing. So always bear that in mind when identifying twins. There's a hell of a lot to digest there, I understand. I hope I have not overcomplicated it, made it nice and simple. I will be posting this slide on my Instagram, so you can always refer back to that or this video of Rob's. The next bit of footage coming in now, guys, it's sped up slightly. This is the steps and procedures that I went through to save the healthy twin. It was obvious by this point the deformed twin was not going to make it. It is quite gruesome at some parts, so you more squeamish viewers, you might want to click away for the next couple of minutes, but I do advise that you persevere with it. I'm so chuffed that I captured this footage, and I really believe that there's some beneficial teachings within this clip. So persevere with it, guys, and I hope you all enjoy and get something from it. So, what went wrong? If these are in fact identical twins, why is one genetically deformed and one is not? The first conclusion that you might jump to is an incubation error. I thought this up first too, but given the fact there was two healthy siblings hatch out of this clutch which were subject to the same incubation temperatures and methods, it does render that conclusion a lot less likely. Myself and Mr. Barrowcroft have discussed this at length in the past, and we believe a lot of ball python defects are attributed to an incubation error, but there are many other things that can occur genetically which will cause these kind of problems, like parthenogenesis. There are actually, believe it or not, two kinds of parthenogenesis: the female form, which is referred to as gynogenesis, and the male form, androgenesis. Gynogenesis being a purely asexual form of reproduction where the female's DNA is duplicated to create a diploid cell. With androgenesis, it happens in exactly the same way, the only difference being it happens inside a female's egg cell, but the female's DNA is disregarded, and it's the male's DNA which then duplicates itself to create a diploid cell. Whenever we have one of these parthenogenic events occurring, the probability of that single parent having a defective gene or multiple defective genes along its DNA strand are actually very high. So problems occur when you get parthenogenic offspring where it's making a copy of one side of its DNA. So wherever that defective gene is, you're getting two copies on either side of the allele. So you're essentially ending up with a homozygous defective gene, nearly always resulting in a deformed hatchling or a hatchling that never develops in the egg. Although very interesting to me, it doesn't explain why we ended up with identical twins and one of the twins was deformed. So then I'll take you to this diagram where we're seeing a good image of how the fraternal twins are formed. You can probably see a lot more clearly there why I refer to that as two eggs within one egg. But we are interested in the identical twins. So sperm fertilise the egg. We now have one half of each parent's DNA fertilising that egg cell. It's at this point here where the fertilised egg cell now has a haploid from both parents, so it is now a diploid cell. It already has the blueprint to determine the gender of that animal. This happens in our snakes a short time after ovulation, which typically occurs a good 50 days before our eggs even make it into the incubator. So this tells us that temperature sex determination for our incubation temperatures is not possible with our animals, and that the gender determination within ball pythons is genotypical. The fate of the eggs is already decided by the time they make it into the incubator. Then we get up to the juicy bit, the cell division. The cell division is what causes the twinning, and this happens through a process called mitosis. Mitosis just being a fancy word for cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus. This is exactly the same as any kind of tissue growth. If you cut your arm, it scabs over. The skin cells are going through mitosis to repair the tissue. It is at this point here where something went wrong with our Burgundy twins. This could be down to the cell division occurring either too early or too late, perhaps under the incorrect conditions due to temperatures or any environmental factors that I did not notice. There's actually quite a large number of things that could go wrong here, but what we do know is that the deformed twin cell, for whatever reason, the genetic blueprint was incomplete. This snake was literally a few sandwiches short of a picnic, resulting in a deformed hatchling. We did still get left with the one perfectly formed hatchling, albeit a little small. Luckily for him, he was born into captivity, so we can do a few little things to help him along the way after receiving a little bit of a shaky start. Thank you guys, I hope you all enjoyed that. You may tell from the footage I'm at the absolute ceiling of my knowledge there. And I don't want to talk about things that I don't understand. So for a little bit of a deeper understanding, we'll go back to Rob and Mr. DNA for a more comprehensive breakdown. Thanks very much again to Rob for having me on his channel. It is a privilege to be invited onto such an educational channel, and that's how we wanted to tackle this topic, is do it in an educational way, so that anyone watching it can have little nuggets of information to take away to further their understanding. Thank you very much again, and I'll see you all on another video.
A DNA strand, like me, is a blueprint for building a living thing.
Alright guys, I hope you are still with us here at the end of the video, and I hope you found that interesting and it whet your appetite for more, because there are 2 more parts in this series. Part 2, the temperature, sex determination and cell division, and part 3, parthenogenesis and genetic deformities. So stay tuned for all of those. Thanks for watching, and special thanks to Joe Tully for his contribution in this video. Outstanding work. Don't forget to share, like and subscribe, and I will put links to Joe Tully's channel down below so that you can share, like and subscribe on his channel, and we'll see you next time.